Okay, that developed in bacteria, and that chromosomes, when that chromosomes develop, that mitosis, that is cell divisions, came in name of that of mitosis. But before that of mitosis, there was a cell division. There was a type of cell division that is called a mitosis. Okay, now we'll have to know about that of a mitosis. Okay, so that you now see that of a mitosis. First, you will have to discuss about that of oldest type of cell division. That is the most ancient type of cell division. That is mitosis, and it was basically found in that of bacteria. Okay, in that bacterial cells, that such type of cell division occurred. So that is mitosis. First, you will have to discuss about that of mitosis. Okay, mitosis. This type of cell division was first coined by that of Remak, and that first such type of cell division, that is mitosis, was first discovered by Remak. It was first discovered by that is discovered by that is Remak. Okay, was first discovered by that of Remak, and later described by Fleming. Later described by Fleming. Okay, Fleming. So that means that mitosis, which is the oldest mode of cell division that was found basically in that of bacteria, was first discovered by Remak, R-E-M-A-K, Remak, and further it was described by Fleming. Okay, so mitosis discovered by Remak and described by Fleming, and it is a such type of cell division, students. No need of chromosomes, no need of spindles, nothing, no mitotic spindle, no need of that of microtubules, nothing's needed for that type of cell division. It is a unique cell division without the help of chromosomes. Okay, without the help of chromosomes, that means such type of division occurs in that cells of bacteria. And only thing that you will have to remember, that there is a constriction developed first of all in that of a DNA. Okay, in that of nucleus part, that is constriction. Occur, and after that, that constriction lead to division of that of nucleus in that of two types, in the two parts, and after that, then cytoplasm also constrict, and that also divided into two parts. Okay, then you will have to see that mitosis was discovered by Remak and described by Fleming, and is often called the direct division. It is also called the direct division. Okay, direct division. That means no need of a mitotic spindle, no needs of chromosome. The mitosis is a type of cell division where there is no requirement of mitotic spindle or that of chromosomes. Without chromosomes, without spindle, such type of division occurs. Owing to that reason, it is called direct division. Okay, it is called, so mitosis is called direct division. You know that mitosis is called, that is equational division, that is equation. Mitosis is a direct division, and it is occurs without the formation of spindle, and of course without the formation of spindle, of course without the formation of spindles, as well as chromosomes. Okay, chromosomes. So that it is a direct division and occurs without the formation of spindle as well as chromosome. For the division of a nucleus, that means in mitosis, how it is a process, that means how it is a complex process. But here is that of direct division. Directly that means nucleus will be divided. So no need of any support from that of chromosomes, as chromosomes are not well developed in that of bacterial cells, and that of spindles also. That means that centrosomes are not lacking in that of bacterial cells. So that in that only need of that of that means first nucleus to be divided, after that cytoplasm to be divided. So it is a direct division. Then we'll have to know about that of students develops constriction. First, you see that I'm just drawing it and then draw at the same time. Okay, and you can know better. So you see that this is that of a parental cell. This is a parental cell. Okay, and here you see that this is that of nucleus. Okay, it is a parental cell. Parental cell. This is a nucleus, and this is the cytoplasm. Okay, cytoplasm. This is the parental cell. You mark it that. You mark it that there will be a constriction. Okay, there will be a constriction. You'll see that. Then this cell, you will have to mark that there will be a constriction. Okay, there will be a constriction. You see that this nucleus, this parental nucleus will look like this. Okay, will look like this. Thus means there is a constriction here. So it will look like this. So there is a constriction. So this nucleus will look like this one, constriction. Then after that, this is the stage development. After that, we will have to mark it that they will be divided into that of two parts. Okay, they will have to be divided into. You will have to see that. You see that. Here is a nucleus. Here is a nucleus. Then this cytoplasm, that you see that there is a constriction in cytoplasms. First, you see that constriction in nucleus occurs. After that, constriction in cytoplasm occurs. So that in that case, that you see that two nuclei. That means this constriction. After constriction, that nucleus divided into two nuclei. Then you see that there is a constriction in cytoplasm. This cytoplasm looks like this. Then after that, there will be two. Then after that, what will mark? There are two daughter cells. Okay, that is two daughter cells. That is called the mitosis. That is called that of mitosis. Okay, that you see that first parental cell, cytoplasm. That is the normal parental cells. Then we will mark that constriction occur in the part of nucleus. That is constriction marks in that of nucleus. So it looks like that of two parts. Then these two parts will segregate from each other, and they will have to maintain a separate identity. And at the same time also constriction will be marked in that of cytoplasm. We mark that there is a constriction in that of cytoplasm. After that, these two parts will break from each other and will have to be two daughter cells. So you see that there are two daughter cells. So this is called that of mitosis. Okay, mitosis. And mitosis is generally found in that of bacterial cells, parameciums. Okay, it is generally found in that of bacterial cells. Okay, that is also protist parameciums. In that of paramecium. Okay, it is also found in that of bacterial cell, parameciums, and that of diseased and old tissues. Diseased and old cells. Okay, old cells. Old cells are tissues. You can have to tissues also. You can. Okay, so that these are found in bacterial disease and old cells that chromosomes not function properly in that old and that of disease cells. Okay, in that of disease and old cells or tissues. In that cells or tissues, that in absence of that of malfunctioning of chromosomes don't functions in that case of disease and old cells or tissues. So in that case, that means there is a constriction occur in that of nucleus of the cell. Then it is divided into two parts. Then constriction also occur in that of cytoplasms. So it is also divided into that of two parts and separated and form that of daughter. This is the daughter cells. Okay, this is that of this is that of daughter cell. Daughter cell. That is daughter cells. Okay, so this one is that of daughter cells. So that things happens. So mitosis basically found in bacterial cell, paramecium and disease, old cells and tissues. This is not in a course, but I have given you little bit more. That is idea. So just only 5 to 10% will have to little bit know, because to have to gain that of complete knowledge about that of cell division. If you know about that of mitosis, anyone will ask that what we mean by mitosis, then there will be certain loopholes or that weakness on your part. So in order to make the complete knowledge, so that I have told about that of mitosis. Okay, okay, students. Then I'll have to ask you that of mitosis. Now you are ready to give the answer to my question. Okay, first questions I'm asking you that who first, that is first of all, who discovered that of mitosis? Anyone? Very good. Who described it?
Fleming.
Fleming. Very good, Fleming. And that means in which cells that type of cell division generally found?
Bacterial cells, paramecium, and disease fold cells or tissues.
Yes, disease and old tissues or cells or tissues. Very good. And that means what happens in that of nucleus, that it makes into two units.
A constriction occurs.
Undergoes constriction and
First constriction in mitosis. First constriction is found on which part?
on the nucleus.
Nucleus. Very good. Nucleus. Then
Sorry,
Cytoplasm.
from them two tissue separate.
Very good. Very good. Brilliant. Okay. So that now we'll have to know about that of the most important one, that is mitosis. Okay, students, you will have to know about that of mitosis. 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 Okay, mitosis. First of all, we know about that of mitosis. What about that of first described by, that is Edward Strasberger. Okay, German biologist. First described by German biologist. Okay, Edward Strasberger. That is Edward Strasberger. Okay, Edward Strasberger. Who described it? German biologist Edward, that is Strasberger. Okay, and in the plant cells, he described it in plant cells. In plant cells, you have to remember that two names. First described by German biologist Edward Strasberger, in which cell? In that of a plant cell. Later by another. Okay, later by another German biologist. Okay, another that is first in plant cell, it was described by Edward Strasberger, and then by German biologist Walter Fleming. Okay, that is Walter Fleming. Okay, Fleming. The first it was in animal cell. In which cell? In animal cell. That you remember, students, mitosis first described by German biologist Edward Strasberger in plant cells. Later, another German biologist Walter Fleming described it in animal cell. In which cell? That is in animal cell. Okay, animal cell in 1879. In 1879. Before that, No one knew about that of mitosis. Mitosis is a type of cell division that occurred in animal cell. No one knew about it. It was first described in 1879 by German biologist, that is Walter Fleming. So you have to remember that first described by German biologist Edward Strasberger, then followed by German biologist Walter Fleming in animal cell. Plant cell and animal cell, two biologists, that means get that described about that of mitosis. Okay, that point you have to remember. That means who described it. Then we'll have to know about that where that mitosis takes place. Mitosis takes place in that of somatic cells, not that of sex cells. It is developed in that of somatic cells. Hence, it is called somatic division. Takes place in that of somatic cells. Going to that region, it is also called that of somatic cell division. Okay, somatic cell division, that is mitosis occur, that is somatic cells, and that is called that of somatic cell divisions. Okay, somatic cell divisions, and it is occurs in the, that means it is also in plant meristematic tissue. That in animal cells you will have to find it somatic cell in animals, and that is meristematic tissue in plants. In plants, it is found in that of meristematic tissues. Okay, meristematic tissues, and also some roots. Okay, growing roots. That is even that of leaves. Okay, even that of that is buds. Also, that type of mitosis occurs. So it is found in that of meristematic tissue in plants. Basically, it is found and somatic cells in that of animals. Okay, so mitosis occurs in which part? That is, you have to remember that is somatic cells of that of animals, and as well as that of meristematic tissue in that of plants. That is occurrence. Okay, that is occurrence. It occurs in that place. Then we'll have to know about that. That means it occurs. That means last. That means it is the average time. That is average time it has been taken for the mitosis is that of that means it takes that last on an average from 30 minutes to 3 hours. It takes average on average basis, average time for mitosis is 30 minutes. Is 30 minutes to 3 hours. 30 minutes. Average time of mitosis is 30 minutes to 3 hours. That means it will have the minimum times, maybe have 30 minutes to that 3 hours. So it has taken the 30 minutes to 3 hours. It is time taken. Some mitosis, some organisms, the mitotic cell divisions occur within 30 minutes, and there are some organisms. That means mitotic divisions is the longer ones, and it takes in that of 3 hours. Okay, but in case of a human being, it is for that of 1 hour. Okay, that is 1 hour. So that the average time 30 minutes to that of 3 hours, that you have to remember. And it is the division of parent cell into two identical daughter cells. Okay, it is the division of parent cell into two identical daughter cells. And so that it is called that is equational division. So it is called that equational division. You see that it is a parent cell. Okay, and having that of nucleus, and it will have to be divided into two equal daughter cells. That means two daughter cells are equal. Two equal daughter cells having same volume of cytoplasms, having same nucleus, having equal DNA materials, everything's equal. Everything equal. So into that region, mitosis is also called that of equational divisions. Okay, you see that. This is the parent cell. This is a parent cells. And that parent cells divides into two equal daughter cells. This is two equal daughter cells. Two equal daughter cells having everything same. Whatever been the first daughter cell, second one is the same is also in the second daughter cells. So nothing varies here. Nothing varies. It is completely equal. Going to that regions, that means they are most of time that of you see that plants or that of some that is of micro organisms. They look like that of in similar in their shape and structure. It is going to that of that is mitotic division. So it is occurs equally. That means that is true. That is everything same. Volume of cytoplasm here, that's same. That is also DNA amount that is equally distributed. Everything same that you have to remember. Going to that region, it is called that that is equational divisions. Okay, that is called. So mitosis is called that equational division. Going to that region of that equal distribution of in that is shell compound, that is equal distribution of that means amount of cytoplasms, that is DNA, amount of DNA, that all everything that will be equally distributed among the two, that is cell organelles. So that means you know that yesterday I had told you that in that gap 2 phase that how everything is becomes doubles. Okay, yes, that is very good, Aditya, that all the things are equally distributed. Very good. Going to that region, they are called that for two equal daughter cell and equational division. That point you have to remember, student, that I have told you that direct division. Mitosis is a direct division, whereas mitosis is an equational division. Okay, that point you have to remember. And that means it is an elaborate process, students. Mitosis is an elaborate process. It is a long-run process. I have told you that in some cells that it happens within the 30 minutes, and some cells also it happens that for 3 hours. So it is an elaborate, complex process. It's not easy one. That mitosis is an elaborate process which involves a series of important changes in the nucleus as well as cytoplasms. You can have to see that. That means a series of change in that of nucleus. For the division of nucleus into the two equal parts, you mark that how in that of prophase or that of in metaphase and that of anaphase, telophase. That means what that is mitotic spindle and that means or centrioles. What they play the important roles for that of divisions of that of chromosomes into two equal parts, and then division of that of nucleus into two equal parts. How they are, that means doing pivotal works, and so that how it is a complex process for that division of that of nucleus and division of chromosomes in equal way. So it is a long run process. It is an elaborate process. It is a complex process. Okay, so it is not an easy process, that mitosis. And then you will have to know that mitosis, okay, includes the main events. That is, it includes mitosis includes main events. Yesterday, students, I had told you that two main events includes in the mitosis. Have you remembered, students? Yes, Jyoti?
Yes, sir.
Have you remembered that two important process occurs in mitosis? Aditya?
Yes, sir.
That yesterday told that karyokinesis and that cytokinesis.
Echinasis. Yes, sir. Yes.
Not to remember that mitosis, that are two important phases of mitosis. Mitosis has also two important phases. That is, one is that of that is karyokinesis. Another one is that of cytokinesis. Okay, that means in karyokinesis there are four phases. There are four phases. First one is that of prophase. Okay, first one is that of prophase, then metaphase, then anaphase, then telophase. Okay, telophase. That means mitosis occurs of the two events. Okay, there are two important events. The two main events occur in mitosis is karyokinesis and cytokinesis. Karyokinesis means that is a division of nucleus. Cytokinesis means division of cytoplasm. That is the division of nucleus. and this is the division of cytoplasm. Okay, so that is first nucleus will be divided, then cytoplasm will be divided. You have to remember whatever I am telling to you. That means on that day also you have to revise yourselves and remember. So that karyokinesis is of 4 phases. That is, there are 4 phases found in karyokinesis. For division of nucleus, that means 4 phases are required. Okay, that prophase. This one is the prophase, then metaphase, then anaphase, then telophase. This is the first phase of karyokinesis. First phase of karyokinesis is prophase, then followed by metaphase, then anaphase, then telophase. Okay, and this prophase generally it is that of a lengthier phase, and it takes 30 minutes. For that of a human being, that is prophase takes 30 minutes. Okay, 30 minutes. Then that metaphase take that of 3 to 10 minutes. 3 to 10 minutes. That metaphase. And the anaphase is the shortest phase. It is a short. Prophase is some a difficult process. It is a difficult phase. Some is complex phase. But metaphase is a simple and short phase. It is 3 to 10 minutes. But whereas anaphase is the shortest one. Okay, that anaphase is the shortest one, and it takes 2 to 3 minutes only. And that telophase is some way that it takes to that of 20. Okay, that of 20 to 25 minutes. 20 to 25 minutes. It is also some sort of that is like that of a prophase. It is also some sort of a complex process. But metaphase and the 2 middle phases, metaphase and anaphase are the short phase, whereas metaphase takes 3 to 10 minutes, but anaphase is the shortest phase. It takes only 2 to 3 minutes. So this is about that of karyokinesis, prophase, metaphase, anaphase, telophase. Now we'll have to discuss one by one after another. First, you'll have to know about that of prophase. Then we'll have to go to that of metaphase. Then we'll go to that of anaphase. Then last phase is that of telophase. Okay, if time that means possible, then I'll have to tell you cytokinesis, division of cytoplasms. What that means in which type of the cytoplasm that is division cytokinesis found in that of plant cells and animal cells. Okay, but if not possible, tomorrow I will tell cytokinesis and that of importance of that of mitosis, by which you'll have to conclude the topic of mitosis. Then we'll have to proceed to that of meiosis. The next Thursday that will be test, and it will cover that of cell cycle and cell division. All questions, whatever be, I'll have to ask you after the end of my lecture. That will be the questions in that of first day. So you should be carefully do it, and you should have to make no mistakes in first examination. On the classes you make mistake, no problems, but in the examination you must have to do all the 10 questions. All the students who are attending the by classes appear in the examination should have to make all 10 questions correct. Okay, then we'll have to proceed to that of prophase, metaphase, anaphase, and telophase. Most important students, you have to remember it. I will have to give you notes. Yesterday I could not send it. Today I will send after just over my class, and you will have to know about it, describe and understand that of notes, and understand here and after the notes, and if doubt, then you ask me. Okay, then we'll start from that of prophase. That prophase, students, you will have to know about that of prophase. It is the first stage of prophase. It is the first stage of cytokinesis. Sorry, that is karyokinesis. It is the first stage of that of first stage of karyokinesis of mitosis. That is, I'm just discussing about that of prophase. Okay, prophase. That is first phase. Prophase, the first stage of that is karyokinesis. This is the first stage of that of karyokinesis. Okay, that is first stage of, you know that I'm just noting. It is the first stage of karyokinesis of mitosis. That point you have to remember. General asks, in question. Which is the first stage of karyokinesis in mitosis? Answer will be prophase. Okay, answer will be prophase. So that point you have to remember. First of all, students, I am telling you that means in the gap to phase of the interphase, that means all the cell organelles are doubled. So there are two mitochondria, two. That means for which regions? That means cell will be divided. Division will take place. So every preparation is being made in that of interphase. It is not a resting phase. It is that is energetic phase. Okay. Now that M phase is called that of most active phase, but resting phase. But that of interphase is not not inactive phase. It is also active to some extent because preparation good means result is within our reach. Okay. So for that means you will have to realize in that basis how important is that of interphase. In interphase, what happens that you see that chromosome numbers never be doubled. Okay, chromosomes number never be doubled. But they are in chromatids. That will be in M phase will be chromosome same. That chromatids make that of a new copy of it. A new copy of it, it just makes. It is going to that of a doubling of that of a amount of a DNA. Doubling of a amount of a DNA. That means you see, simply you see that this is just a chromatid. Okay, it is a chromatid. That is chromosome is found in chromatid in that of a interphase. But in that after that synthesis of a DNA, that is synthesis of a DNA, you will see that what you will see that there is another. That means you will see that means this is a part. This is a chromatid. Another chromatid will also develop. This one is a chromatid. This one is another chromatid. So this is a chromatid. This is a chromatid. This one is a chromatid, and this is another chromatid. Okay, another chromatid. It is just a replica of that chromatid. It is going to the doubling of the amount of a DNA in S phase. In synthesis phase, the 2Na, the amount of DNA becomes that of 4Na, so that this chromatid will also a replica. That means it will also another replica. But chromosome is one. It is a one. But there are two chromatids. Here what you are finding? Here you are finding that of two chromatids. Okay, two chromatids. But here it is a one chromosome. It is also one chromosome. One chromosome. Chromosome number is one. Chromosome number is one. Chromatid numbers two. And these are the four arms. These are the arms. This is the arm. This is the arm. Okay, you see that. You see that. You see that. That means this small one. This small arm is called PR. Small arm is called PR. The long arm is called QR. You should have to remember this concept because for the examination point of view. In that of chromatids, you see that this is called the small arm. P arm is the small arm, and this one is the large arm. And there is that centromere. It is a centromere. Here is a centromere. That centromere, okay, that centromere join the chromatids together. This is a what? This is a, that is, we mark it that this one is a centromere. Okay, this one is a centromere. Centromere. And these are the arms. There are four arms. How many arms are there? Four arms. Okay. How many centromere is there? One centromere. One centromere. Okay, centromere. And that is kinete force. That you see that there are two kinete force. You see that here is a chromatid, that is a kinete force, and you see that there is, you see that you mark it that there is another kinete force. Okay, this one is two kinete force. That kinete force, that is are two in numbers. There are two in numbers. Okay, there are two in numbers. That is centromere one, but kinete force are two in numbers. There are two in numbers, four arms, and one chromosome. One chromosome, two chromatids, four arms, one centromere, and two kinetones. So you have to remember that one in a chromosomes. Okay, and this part, if this part is segregated, that means it will be chromatid. This part will be chromatid. So after cell division, that will be divided into two equal parts. Okay, that idea you have to just remember in that of chromosome. That is chromatid. This is also chromosome. That is also one chromosome. This is just a one replica of that of chromatid attached to that of chromosomes. Okay, and this one you have to remember that these are the sister chromatids. Okay, that are sister chromatids. They are two sister chromatids. That you have to remember. Now come to that of prophase. Students, I have already told you that prophase is that of first stage of that of karyokinesis of that of mitosis. And you see that what happens in that. I'm just drawing, and you also draw with me. Okay, you see that you see that there are you see that these are the chromosomes. You mark it that there are the chromosomes. These are the chromosomes. These are the chromosomes. Okay, and chromosomes, you know that the centrosome, if centrosome, you see that this is just, that is, if centrosomes are on that a perpendicular basis, this will be called as that of centriole. You see that is a centriole. So you can have to mark it that. You can have to mark it that here is a centriole, here is a centriole. So centriole synthesis takes place in that of SH, and that centriole come to that of a polar region. That is in the polar part, centrioles. Okay, and centrioles will develop. You see that centrioles will develop. That is the centriole that will develop that of a microtubule. These are that of a microtubule, and these microtubules are called that of asters. Okay, this one is called the asters. These microtubules are called asters. Okay, are called aster, and this one is that of that is centriole. Okay, centriole, and here also that of you see that there are also that of microtubules, and these microtubules are called that of asters. So there are two asters. Okay, there are two asters. You see that here is that of that is, these are the chromosomes. The number of chromosomes here are 4. Okay, number of chromosomes here are 4. The number of chromatids here will be, students, how many chromatids will be here?
Sir, 16.
What the numbers are 4. How many chromatids will be there?
8.
8. Very good. Why you are fearing?
8.
That is from Tusharanjan. Not do what you are chromatids. Are how many chromatids? Each chromosome 2 chromatids. Yes, that is Aliba. That is right answer. So there are number of chromosomes. I'm just showing you that there are so many chromosomes there, but number of chromosomes are 4. Number of chromosomes here are 4. Okay, and that of number of chromatids will be 8. Okay, and this was the centriole. Those centrosomes are on that of perpendicular. Two centrosomes are on the perpendicular basis, so they are called the centriole. Centriole will go to that of polar regions. They will develop microtubules. These are the microtubules. These microtubules are called asters. Okay, asters, and then that of spindle formation will take place. Okay, the spindle formation will take place. You see that. That means if a spindle will go independently, this one is called continuous. This one is called that of a continuous race. Okay, these are the continuous spindles race. This one is the continuous. That is continuous spindle race. Okay, and you see that if anyone comes within that of chromosomes, okay, within that of chromosomes, this one is called that of a discontinuous spindle web. This one is called that of a discontinuous, okay, discontinuous spindle webs. You see that? I'm just telling you that you understand that. That means these rays, that means these rays, you see that these spindle fibers, these fibers will develop. Here is that. You see that. This is a, that means you see that this is a chromosome. There are two chromatids. How many kinetophore will be here? Students, how many kinetophore will be here? One or two or three or four?
Sir, 4 in total, 4.
Sir, 4.
No. How many kinetophore will be there? I have
Sir,
told you here, I had shown you that
two.
that is one chromosome, two chromatids, four arms for one chromosome, four arms, one centromere, and another one is that of two kinetophores. Okay, you see that. You can have to see that. That means it is a one kinetophore, another one will be. So that is centromere will be one. That means which are joining the two, that is two chromatids, but there are two kinetophores for each chromosomes. Each chromosomes. Here you see that. That means here you see that. That is one rays come from that of suppose this is the north one, this is the south pole. From that of north one, you see that from that of north one that is one. You mark it that one thread. Okay, one fiber comes and comes, and here you will see that it will have to touch that of one kinetophore and will go. Another will go from that south pole. That also meet that of kinetophore and they will have to go. So there are two types. So there are two kinetophores, whereas one takes the thread, one takes the fiber of south pole, and another kinetophore takes the fiber of that of a north pole. Okay, so that one in one chromosome there are two kinetophores. So that one will have to take that of fiber, will take the fiber of north pole. One will have to take the fiber of that of a south pole. Okay, south pole. So like this, you should have to see that. That means this is also, this one also joins, and this one also, that you can have to see that. Okay, like this way, like this way. Another one you see that it will go through this way. Okay, go through this way, and this one is the continuous without any obstruction, without any chromosomes or kinetophore. They are, if they are spread, they are called that of a continuous. These are the continuous. Okay, these are the continuous. But those are coming within that of chromosomes, they are called that of a continuous, discontinuous. Those are coming through that of chromosome, they are called discontinuous. Those are freely coming, they are called as that of a continuous. Okay, continuous. So in that case, what happens? You'll have to know that. What are the major events occur in that of a prophase? You can have to know that. First point is that of a chromosomes, condensation of chromosomes. That means you see that chromosomes, that is chromosomes come from that directly from that of gap 2, and that are also in a doubling manner. So you see that replica also comes, so they will look like this. And but the condensation will take place. Condensation means that water will remove from them. So, okay, water will be removed from them. So what they were looking like this. But after the condensation, okay, after the condensation, they will look like this. Okay, they will look like this. They will look like this. Okay, after condensation, so the water amount will go away from their body parts, so they will look like this. Okay, that is called condensation. You see that this is called condensation, and they will more distinctly be visible. Okay, you see that they are distinctly that visible and going to that of a condensation. So that is condensation. Condensation means, that means water vapor will go away and they will be in condensed form. So they will, so that chromosomes will look like this way. Okay, will look like this way, and that will have to find that chromosomes are seen to be composed of two chromatids. I have already told you that chromosomes will seem to be attached with two chromatids. Okay, two chromatids, And same thing what I have told, that will be found in that chromosomes. One chromosome will have two chromatids, four arms, one centromere, two kinetochores. Okay, all these will have the same things. So that will have to be found. After condensation, chromosomes will look like this with all four arms and one centromere, two kinetochores, and that of two chromatids. Okay, two chromatids. That is condensation of chromosomes. And then that is centrosome which are undergone duplication during interphase moving toward. You see that? That means which that you see that that is centrioles that centrosome which will have to be on perpendicular basis. These are called centrioles. That centrioles after the S phase reaching the G2 phase and will reach the prophase, and that will have to move to that of central position. So centriole will go to that of central position, and that of a condensation of chromosome. First event, condensation of chromosome. Then second event is that centriole will move to that of that is polar position. Okay, they will have to go to that of polar position, and that each centrosome radiates microtubules called asters, and the these radiate, those who are looking that they are called radiate microtubules. These radiate microtubules are called that of aster. So aster. Okay, so there are two aster. This one is the one aster, and this one is that of another aster. So another aster. So the two asters together with spindle fibers. So these spindle fibers and that asters are called that of a mitotic apparatus. Okay, these are called. So that of a spindle, that is spindle fibers. That is spindle fibers plus that of asters. Two asters are called that of mitotic. That is apparatus. Okay, apparatus, mitotic. So this one is called mitotic apparatus because this one is one aster, this one is one aster. So that centriole along with that of microtubule fibers are called asters. These are two asters, and these are that of that is spindle fibers. So spindle fiber and that of asters are called that of mitotic apparatus. So that mitotic apparatus is also two mitotic apparatus are also made. That is mitotic apparatus are made after the addition of that of spindle fiber to that of two asters. Okay, two asters. So this one is that of prophase, in which what you will see that first event is that of condensation of chromosome takes place, and that means dehydration process. So water will go away, and then after condensation that chromosomes will be to some extent be visible, and then that of centrioles will go to that of polar regions, and they will have to develop that of microtubules, range of microtubules. That range of microtubules along with that of centrioles will be called as the asters. So there will be two asters, and that two aster will form that of develop to that of spindles, and that spindle fibers, that spindle will have to develop that of fibers. One fiber will, that means those will join to north and south poles. They will be called as that of continuous spindle fibers because they join without any obstruction, and those will come inside that of chromosomes. They are called that of discontinuous spindle fibers. Okay, and then we'll have to know about that of that means if one fiber comes from that of north pole and joins one kinetochore, then another will have to toss that of a second kinetochore. So in this way, that of spindle formation is made, and that means in prophase, that means chromosomes are being ready. Okay, chromosomes are being ready for that of a metaphase stage. How they will have to move to that of meta, and in this phase, that means Golgi apparatus, nuclear membranes also not found, Golgi apparatus also not found. Okay, not found in this case, and nucleolus also not found in that of prophase. In that of prophase, cell organelles, that is some cell organelles are not found, and some cell organelles are not found. So it is easy for that of spindle fibers to come to that joining that of two asters. Okay, joining of the two asters is possible due to the absence of that of that is some organelles. Okay, some organelles in that of the center of that of cytoplasm. Going to that regions fibers. Okay, development of fibers takes place. So this is about that of that is prophase. Okay, in prophase you have to remember condensation of chromosomes takes place and centriole moves to that of that is opposite poles. Okay, opposite poles, and then nucleolus child, that is nuclear, that is nuclear membranes not found. Nucleolus Golgi apparatus also not found in that of a prophase. This is the main important events that we will have to remember in that of a prophase. Okay, let's come to that of a metaphase. Metaphase. We'll have to discuss about that of a metaphase. Metaphase, students, is that of the shortest phase, but metaphase is such a phase in which you can have to determine and counting of the chromosome numbers, counting of the kinetophores, counting of that of centromere, all are possible, all are visible, all are possible in that of metaphase. Okay, that means for that of chromosomes, determination of chromosome size, numbers, etc. is possible only in that of metaphase. That concept should be very clear. That counting of the chromosome number, their centromere, that is chromatids, number of chromatids, etc. can be learned only in that of a metaphase. So determination of chromosomes arithmetic is possible only in that of a metaphase. And a metaphase is a short phase. I have told you that it is a short and a simple phase which have only 2 to 10 minutes, which only take 2 to 10 minutes. And then prophase takes that of 30 minutes. Then come to that of metaphase. Okay, we'll have to discuss about that of metaphase. Metaphase takes that of that I have told you that it takes only just that is 2 to 10 minutes. Metaphase takes 3 to 10 minutes. 10 minutes. And it is the most important phase. Okay, it is the most important phase. And you see that I'm just drawing about that. In metaphase, you can have to mark that. Okay, in metaphase, you can have to mark that. That means all that of chromosomes will come into that of equator. Okay, that chromosomes will station at the equators. You see that chromosomes are centers at that of equators. Equator. All chromosomes which are in the different position of cytoplasms will come to that of equator in the middle position, to the middle. It is the most important event of that of metaphase. And you see that this one also one that of centriole, another centriole, also that of development of that of asters. Okay, that is microtubules. Also, you should have to see that of microtubules. Then you see that. That means the development. This one. This one is called that is continuous. Okay. This one is that of continuous. That is continuous fiber. And this one is called, which one comes in that? This is called that of what? This one is discontinuous. Okay, discontinuous. Discontinuous like this. The main point you have to students remember that chromosomes which are condensed at a different location of that of a cytoplasm in that of a prophase, they will come to that of middle position, will come to the equator. Okay, equator. So they will have to make a equatorial plate. Okay, they will have to make a equatorial plate. Equatorial plate. So they are making plate-like structure. That is metaplate-like structure. That is called equatorial plate. This is the equator, the middle part. In that middle part, all the chromosomes will be, that is, will have to be attached. All that will attach at the chromosomes will be attached to the equator. So they will form a that is a special plate. That is called what? That is called metaplate. So it is a metaplate. At the metaphase, a plate-like structure is found because all chromosomes come to that of middle position, come to the equator. So it makes like a plate-like structure. And another thing is that it is the stage where morphology of chromosome is most easily studied. That is chromosome. Okay, the study of chromosome. The study of chromosome is the most important. Morphology of chromosome. Morphology of chromosome. Morphology of chromosome is easily studied. Is easily studied in this phase. In this phase, you can know about that all of these things. That is all of it will look like this in that of prophage. But the counting of arm centrosome, all that is centromere or that of chromatids, everything that can be possible only that of a metaphase. So morphology of chromosome is easily studied in this phase. And that of metaplate, that is metaplate, development of that of metaplate. Okay, metaplate will have to develop in that phase. That is phase alignment of chromosomes. That is metaphase plate. That is metaphase plate. That is metaphase plate. Metaphase plate. So main thing we'll have to remember that metaphase, that all chromosomes come to that of equator and the metaphase plate is formed, and the morphology of chromosome is easily studied in this phase. That's important is for that of metaphase. Okay, only that things you have to remember. Okay, only things you have to remember, and that means and the kinetics will have to be seen that is in that phase, and these are the complex protein. These are the complex proteins. What are the kinetics? These are the complex proteins that you have to remember, and they are 2 in numbers for the each chromosome. They are 2 in numbers. That things is about that of metaphase. Only metaphase things you have to remember. Morphology of chromosome easily studied, and all the chromosomes comes into that of middle equator levels, and they will form that equatorial plate or that called metaphase plate. This one is also called equatorial plates. Okay, equatorial plate. Okay, this one is also called equatorial plate. This one is called metaphase plates. That you have to remember. Sometimes question asks, equatorial plate is found in which phase of mitosis? Answer will be that of metaphase. If the metaphase plate comes, anyone can have to answer. So question will be that of equatorial plate. And the morphology of chromosome easily studied, that is in metaphase. Okay, that's about the metaphase. It is very simple. It is very simple and short, which takes only 2 to 10 minutes. Then students come about that of anaphase. Okay, about that of anaphase. In anaphase is also very short. It is the shortest phase. It is the shortest phase and the most simple phase, which lasts anaphase. That is anaphase. You see that of anaphase. Anaphase is the most, that is shortest phase and the most simple, that is simplest phase, and it is just take 2 to 3 minutes students only. Only 2 to 3 minutes it takes. Okay, and in this phase you will see that means those chromosomes aligned at the equator, they will be separated. Okay, they will be separated. We will have to see that chromosomes will look like this. Will only look like this. Okay, the chromosomes will look like this only. Will look like this. Only one chromatid you can have to find, because chromosomes will be, the 2 sister chromatids will be segregated, will be separated in this part of anaphase. In this part of anaphase, that chromosomes, the 2 chromatids will be segregated, will be separated. So that will be separated parts of that is chromosome. These are the chromatids which will be separated. And the same thing we'll have to mark it that they are separated. Okay, they are separated to each other. Okay, they are separated to each other and they form that parts of. Okay, and they will have to show it. We see that they will have to just you show it that this is that of centriole, and these asters. You see that they will see that joining to that of that is asters, and they will show like this. They will show like this, and that main event occur in that of anaphase, that is centriole, that is centromere split. This centromere split, so two chromatids separate. So the two chromatids separate, and they will start moving to that towards pole. Okay, each chromatids that is separated. This one come to north, this one go to south. So they will separated part will have to be moved to the different poles. So this one's come to this side, this one come to that side. In this way, they are being separated. So separation of a chromatid takes place in that of anaphase. This is the most important phase. And in this phase, you can have to find that there are different phases of that of chromosomes are found. Okay, that is V shape, that is metacentric, that is V shaped. Okay, then after metacentric, that you can have to find that of that is submetacentric, metacentric, that is L shaped. Okay, then you can have to find that of that is acrocentric, that is J shaped. Okay, then telocentric. The last one is that of that is telocentric, that is I shaped. So different shape of chromosomes can be found in that of anaphase. You can have to see that if you can have to see that is just you mark it that if this is that of if both are equal, you see that if both are equal, that is this is the P arm, this is the Q arm. If P arm and Q arm are equal, this is called that of metacentric, and it will look like that of V shape. It will look like that of V shape, metacentric V shape. If P arm is equal to Q arm. If submetacentric and centromere is in the middle position, if centromere is somewhere, okay, not in the middle position, there will be two arms. The P arm is the small arm, and the Q arm will be that of that is large arm. It is the small arm, it is that of large arm. P arm. So P arm is less than Q arm. In that of submetacentric, it will be L-like structure. And in acrocentric, you will have to it will have to go to that is centromere will go to that of top position. So you can have to find that like this. That means P arm is very smallest and the Q arm is the longest one. So that P arm is also less than Q arm. But in that of that will look like a J shape. Acrocentric is that of J shape. In telocentric, I have already taught it in that of that is cell, the unit of life. I have already taught, but just remembering you that you see that in that of telocentric, that centromere at that that is final position. So there are no P arm, only Q arm is visible. No P arm, only Q arm is visible in that of telocentric. Going to that region, it is called that of I shape. Okay, I shape. So different shape of chromosomes are found in anaphase. Say that centromere split and the chromosomes that is that the separated part of chromosomes, that is chromatids, moves to that of that is opposite poles and attached to that of astral rays. Okay, and then they form that of different forms of that of chromosomes. Okay, that is V-shaped metacentric, submetacentric L-shaped, acrocentric J-shaped, and a telocentric that is I-shaped. In that of that is telophase. In that of telophase, that is also a complex phase. Okay, telophase is that of some sort of complex phase. that is telophase. In telophase, what you will have to find that means astral rays will not be seen. Astral rays, microspindle also disappear. Astral rays also disappear. And that centrosomes, and that you see that means whatever the chromosomes you are find, that will merge with that of nucleus. Okay, that will merge with that of nucleus. So there will be two nucleus. There will be two nucleus found. You see that there will be two nucleus found, and the Golgi bodies and all that of Golgi apparatus to that of endoplasmic reticulums, nucleolus, all are found in that of telophase. Telophase and that chromosomes, that means the nucleolus, that means that nucleus. There will be two nucleus, that means the DNA structure and that of DNA structure will have to, because that division, that means it is the final stage that two nucleus develops, and all the chromosomes that are separated chromatids that have gone to the north side and that to south side, they will merge, the chromosomes will merge with that of DNA and will form that of a nucleus. So there are two nucleus formed in that of telophase. And cell organelles, all cell organelles are also found in that of a telophase. In that telophase, all the cell organelles are found. Okay, nucleolus, nuclear, that is nuclear envelope also develop. Everything will have to develop because it is the final stage of that of that is cell divisions. Okay, so that two DNA structure in that of two nucleus, and everything will have. So it is also some sort of a complex structure. Okay, some sort of a complex structure, and that is, and then the viscosity of cytoplasm decrease, and the shell assumes it is normal forms, and the shell will assume it is normal form when that of viscosity, that is viscosity of that of cytoplasm decrease, so that they will form a normal cell position. So they will have a normal cell. So that you will see that will be divided into two parts. So that cytoplasm, some cytoplasm, that so there is a division occur. So that will make into a two. But that is about that of karyokinesis. That is that last stage of karyokinesis is the telophase, in which that two nucleus develops. But cytoplasms will be developed into two equal parts. Only students are that of a cytokinesis. Okay, and tomorrow I will have to teach you that of cytokinesis and that of that is important significance of mitosis. So today students, that is 11:15. So I have taken that 15 minutes class. Okay, so I can do any question today, but next class will start and you will have to take rest. But sadly that I have not given any rest for you. Okay, and so thank you students very much for joining the class. Thank you students.
Thank you, sir.
Yes, sir.
Thank you,
Thank you,
sir.
sir.
Yes, sir.
Thank you, sir.
Thank you. Now this is clear. So,